Well, I think we might just get going with our introductions today. So just welcome everybody. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. So good afternoon everyone. My name is Sarah Bennett. I am the Research Coordinator at FightMND, and I'd like to thank you today for logging into the FightMND Scientific Seminar for November. We have a really exciting presentation lined up for you today, which will highlight the critical work of our Australian researchers in the global push to identify biomarkers for MND. So following the presentation today, we are leaving plenty of time for discussions and questions. So if anyone would like to ask anything, please use your reaction button to raise your hand, and then one of our team will enable your microphone and camera and let you know it's your turn to ask a question. Alternatively, you can also type your question into the chat box where our team will be monitoring and read out questions on your behalf. Please note, however, that the chat function is only enabled if you're using the app-based version of Teams, and unfortunately doesn't work if you're using the web-based version. So now to our speaker for today. Associate Professor Mary-Louise Rogers is the head of MND and Neurotrophic Laboratory at the Flinders Health and Medical Research Institute at Flinders University in South Australia. She obtained her PhD in biochemistry at the University of Adelaide, and followed by postdoctoral work at Flinders University. She then established her own independent research group at Flinders, supported by both national and international grants. She leads the Biomarker Collective at Flinders and has an international reputation in urinary biomarkers for MND research. And she's here today to tell us about her journey in MND biomarker discovery and what the future may hold in this space. So please join me in welcoming Mary-Louise today. So take it away.
I'd just like to thank you, Sarah, for inviting me to give this talk today, the last FightMND scientific seminar for the year. And today I'm talking to you about fluid-based biomarkers that are useful to investigate disease progression in sporadic and familial MND. And so, just hold on. So, firstly, I'm going to talk about why biomarkers are important, and what are biomarkers, and what natural history studies, what they can tell us about biomarkers, and why progression biomarkers are important, and how we've actually found some progression biomarkers, and how we now plan to add a fingerprint to enable early identification of MND using biomarkers. So, as you know, motor neurone disease is rapidly progressive, and 90% is sporadic, and around 10% has a familial cause. That means that there's genes that are involved. And in Australia, there's 2 people diagnosed each day, and 2 people die, and most people live 3 to 5 years post-diagnosis. And there's a huge cost to this disease, such as the Deloitte Access Economics report, which actually showed there was, in 2015, it was costing Australia $2.37 billion. And it's actually quite there's a low prevalence in incidence, but it's a very rapidly progressive neurodegenerative disease that has a not great prognosis. So it's often characterised by many things such as limb weakness and fasciculations, which are jumping of the nerves that you can see. Basically, there's a lot of things that occur that lead to this rapid neurodegenerative disease, but we don't really know what causes it, only that there's a genetic risk, which is with this familial MND. And if we look at the history and neurodiscovery of these gene mutations, SOD1, which is around 20% of this 10%, and C9orf72, which was identified in 2011, it's around 40% of all of these familial cases. and then TDP43 or TARDP, and thus slightly less, around 5%, or much less, around 5% of this 10%. And there's lots more gene discoveries. So we're getting to know more and more about what causes this familial disease. We don't really know what causes it. The other thing we do know is that it's a multi-step process. So there's been a lot of work, including by Australian, our own Steve Vukic, which actually showed, following on from Amar Chalabi work, just showed that there's a linear relationship between log age and log incidence, and it suggests there's 6 steps in the Pacific, which agrees with the European study. So Steve Vukic's study agrees with this European study. So there must be some environmental exposure, or 6 genetic steps that actually need to trigger the disease. That's what we know so far. But it's actually quite variable. So sporadic disease can be variable with widely distinctive clinical features, including the site of onset, the age of onset, progression patterns, and molecular mechanisms involved. Even amongst familial MND, there's often this degree of this variability or heterogeneity. So we really need to look at multiple avenues to reduce this heterogeneity, so we can find a treatment that is efficacious. So, in a bit more detail, some of the clinical heterogeneity is quite obvious. So those people that have limb onset, there's around 70% of people, and the 30% with bulbar onset, which actually means people have a shorter survival. There's a variable diagnosis, which means there's a delay in diagnosis, which means there's variable baseline. That's the start of the disease. So, and also the progression rates it influences progression rates. And 15% have this frontal temporal dementia, and the rest don't. The age of onset is variable. There's all these variable things that happen. So one of the things that can actually reduce this heterogeneity is actually using biomarkers. They actually can enable identification of effective treatments and elimination of ineffective. They can reduce heterogeneity to improve the ability to detect benefit. And for familial MND, validation of early diagnosis can enable earlier treatment and also reduce heterogeneity. So what is a biomarker? So the National Institute of Health defines it as a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or intervention, including therapeutic intervention. It's a lot of words, but it really just says that it will show what happens in a treatment and a response to a treatment. So this, I would urge you to look at this FDA working group document, which is called BEST, which is Biomarkers, Endpoints and Other Tools, and actually puts out in quite succinct way what types of biomarkers are and what they are used for. So in response to most often, so we're looking mostly at monitoring biomarkers, pharmacodynamic biomarkers, and predictive biomarkers. But these monitoring biomarkers can be used as pharmacodynamic biomarkers, which I'll talk to you in a minute about. So diagnostic biomarkers, really not useful in MND at present, but could be with an early fingerprint of MND, which I'll talk about in a minute. Monitoring biomarkers, they're measured repeatedly to assess status of disease. They're very important and can be used as pharmacodynamic biomarkers. And prognostic biomarkers, which identify the likelihood of a clinical event, they're very useful in classifying people across the disease. And predictive biomarkers, which identify individuals who are more likely than others to experience an effect from exposure, are also very important in clinical trials, which I'll talk about a bit later. So pharmacodynamic, progression biomarkers and prognostic biomarkers are are really relevant to developing treatments. Pharmacodynamic progression biomarkers and trials and prognostic biomarkers actually help to classify subgroups, in addition to predictive biomarkers, which will also classify patients that are grouped on a prediction of a treatment or prediction of a disease status. So prognostic progression markers in early MND may actually help to find a fingerprint that determines early MND, which I'll talk about a bit later, and reduce the time to diagnosis in early MND and pre-familial MND, and reduce heterogeneity for trials and enable early treatment. So validation of biomarkers for familial MND, which I've just talked about, would enable earlier treatment and pave the way for reducing heterogeneity. As, for example, I just want to put an example of this. An early accurate diagnosis is mandatory for therapies such as the antisense therapies, tofersen, which people have heard of, for those with SOD1 mutation. So that's an example where an early diagnosis may actually improve the effect of a treatment. So at present, the ALS-FRS, which is the revised ALS functional rating scale, is used as the only validated outcome measure in clinical trials. And this actually measures motor decline, and it's based on a total scoring of 48, which is normal. And as people decline, that score goes down. So this has been validated and been used in clinical trials. So prognostic markers are baseline characteristics. So they actually categorise patients by degree of risk. So neurofilament light is an example of a prognostic biomarker. So levels of this at the time of initial evaluation can predict the stage of whether people have more rapid progression and shorter survival. So my colleague in the US, Michael Benitez, had this paper out a couple of weeks ago in Brain, which actually shows what NFL as a prognostic and as a natural history in a natural history trial can actually tell you about MND ALS. So here we've got what happens is NFL supposedly rises in the pre-symptomatic period and early symptomatic, and then it's very stable from diagnosis. But the levels of it at diagnosis will actually tell you whether someone's has faster, intermediate, or slower progression. So these sort of natural history studies are very important, and NFL is a great example of that. So our lab is actually investigating urinary biomarkers. So what are the advantages of these? So most people are willing to provide a sample, and collection is much less invasive than, for example, collecting CSF. That's cerebral spinal fluid, having spinal taps. And also urine is a lot less complex protein mixture and actually much easier to identify, for example, than in blood, which is much more complex. And if you did mass spectrometry, it's much easier to find biomarkers. The disadvantage of you must actually correct for urinary dilution. So we have there is a bit of a debate about whether creatinine is a good correction for urinary dilution. We've been using that, but there's also osmolality. So this is just an example where you can see creatinine versus osmolality, and they're quite correlated. So we do both, but in this instance, I'm showing published work which is all done with creatinine, but we don't believe that using creatinine is a big issue. So progression biomarkers, as opposed to NFL, they actually rise. So if you have a look at say this is some measure that goes up, and if the treatment was to be effective, this slope would go down. in ALS-FRS is opposite, so that it starts off as high and goes low, and you're looking for a change in slope, and that will tell you whether a treatment's working or not. So we've been looking at this biomarker called the neurotrophin extracellular domain. It's actually a receptor, a part of a receptor that's cleaved from motor neuron membranes and other things that actually gets upregulated in motor neuron disease. And as the cell gets sick, the part of the receptor, the extracellular domain, gets cleaved, and this is around 50 kilodaltons and ends in urine. And we started looking at this because we noticed this previous publication in 1988 that hadn't been followed up that said that if you cut a nerve, then you actually see this in the urine after a number of hours. So we just followed this paper and asked the question. So, and what we found, this is a number of years ago, is that we could actually detect, I made a number of monoclonal antibodies, and we could actually use immunoprecipitation and detect the extracellular domain in the urine of MND patients. And by mass spectrometry, we were able to show that it was actually really there in this immunoprecipitated fragment. Published this, and we then developed a enzyme linked immunosorbent assay, and used this to quantify some in this first paper to show that you could measure the P75 and that urine, and it wasn't affected by how much you could easily dilute it out. There was no effect of urine on the measurement of the P75, and it was higher in MND or ALS than in controls. This is just the first paper that we showed, and it was higher in both limb and bulbar onset. So then we went on to a larger study with my colleague Michael Benatar and the Miami group, and then we followed some people for 2 years and measured 2 to 6 times in Adelaide and in Miami, and then we looked at whether P75 ECD was going up, and also whether it was a prognostic biomarker. So the first thing we showed was that in fact it was going up at a known rate, and we're able to quantify this using R and using multiple statistic measurement in R, and showed that it was going up from diagnosis, for example, by 0.29 nanograms per milligram of creatinine per month. And this also occurred whether it was from symptom onset or from baseline, which is the first visit, and it's quite significant. And we also showed that there was a correlation, not strong, but there was a correlation in our group with the group of 54 people between the ALS-FRS and the P75 extracellular domain. And also those people that had higher than 5.4 nanograms per milligram actually had a shorter survival than those people that had low. So but again, it's not strong as a prognostic, but it is. So basically, I guess from this study, it was the most important part of it was to show that it actually did change over time and increase as disease progresses and motor function declines. It was correlated with the ALS-FRS, and also there's correlation over time, which I haven't put here, but it's in our papers. And that is the ALS-FRS over time, and the P75 was correlated as well. So we also showed that the extracellular domain of P75 is predictor of survival, and it was really a world first urinary progression marker for MND. So since then, we really wanted to translate this, and FightMND have been really helpful in helping us to ask the question whether it could be used in clinical trials. So like I've showed this before, so we wanted to see whether there was whether there's a change in the slope. So really we haven't got to the phase 3 trial to look at something like this yet, but we're going step by step. For the first thing we really need to do is actually get a high throughput way of measuring, and this is funded by FightMND, the Ian Davis biomarker facility, and we're able to measure more than one plate a day, which we were doing previously, and reduces error and improves accuracy. And the other thing we did was actually improve our ELISA by having 2 monoclonals, so we previously had a polyclonal antibody, and really for going ahead we had to figure out and have to capture and label both monoclonals to make sure this could go forward. So we have used it in a number of a couple of trials, but I just wanted to highlight one that was probably there's more information on it. Some others there isn't so much information because they were negative trials, but for the Lighthouse trial, the first Lighthouse trial, even though it was a phase 2 trial, we did measure P75 levels, and there was this decline after starting treatment. But really, we can't say much about this because it's not. We haven't had enough people in this trial, and the next part of this trial is actually this phase 3 trial, which is already underway, which has got 390 people enrolled, placebo-controlled, and we hope to determine whether the P75 is a good readout for efficacy. So I should say the Lighthouse trial, which I hadn't explained that well, is actually an antiretroviral therapy. It was developed by Julian Gold, and it's quite that, you know, we don't. So hopefully what it does is reduce this retroviral possibility of having retrovirus re-established or contributing to motor neuron degradation, which is sort of a theory about one of the theories about why how MND starts, but it hasn't really been proved just yet. But there's more and more evidence that these retrovirus might be involved. That's another story. So another type of biomarker we're working on is predictive biomarkers, and this is really about whether we can find a pro-inflammatory biomarker. So when motor neurons are going, they first of all they start getting TDP43 inclusions and such like that, then you have an anti-inflammatory reaction. So the cell is sort of cleaning up all of the debris and things that are going on, but once it gets overwhelmed, there's a switch in the periphery and in the brain to more pro-inflammatory condition. So you get the Th1 response. So, and what happens, you get interferon gamma gets upregulated, and in response to this small molecule neoyotrin is released. That's actually released from in the periphery and from microglia, and it's quite easily detectable in the urine. And so we think it may be a useful predictive biomarker for those in this pro-inflammatory state. So it could be useful for treatments that are targeting inflammation, or to group people that are in this pro-inflammatory state. So to do this, to look at neoyotrin, we actually used a commercial ELISA. And that's a competitive ELISA. It's not probably as specific as the other, like a sandwich ELISA that we actually developed ourselves for the P75, but it was available. So we started with that. What we had is a longitudinal study again, and then we had people that had been measured 2 to 6 times, and we again like with the P75, we compared to the ALF-SRS, site of onset, disease duration. We wanted to ask, is urinary neopterin a disease progression marker? Is it prognostic? And it's so again, it was a study involved with our group and the people from Miami, Michael Bennett's group. So this was published earlier this year, and it showed we showed that actually neopterin was higher in MND ALS than in healthy controls. In the same group, we've got the P75, which is significantly higher. This is not as, but it's probably, if you think that some people are in different inflammatory states during disease, it's probably not surprising. There was a relationship, though, with the ALF-SRS, so that there was, we had some significance, so is it some correlation, and also the P75 and the neopterin were correlated. So what this is showing is that the other thing that we show, I haven't got here, is that actually neopterin is not a great prognostic marker. It is prognostic, but it's not not anywhere the leg of, for example, NFL. We don't think it would be a significant prognostic marker. But what we did show, which I think is great, is that neopterin increases at a rate, and there is a difference between fast and slower progressors, and we're able to measure and have a rate per month, so this is a good result that we can use this rate and in trials. And again, we also measured P75, and we have got virtually the same rate as we had in our previous study. So from the neopterin summary, so urinary neopterin in MND patients is significantly elevated compared to healthy individuals. It's correlated to the disease state. What I mean by that, the ALF-SRS, and it's correlated to the P75 extracellular domain. So it changes at a measurable rate over progression. Now we have two progression markers that could be used as pharmacodynamic markers, one of which could be predictive. So the neopterin is predictive of those people that are in pro-inflammatory state. And there's a podcast here if people are interested, and another article about this important finding in neopterin. So in summary for this part, urinary P75 has been used and actually continues to be used in clinical trials as a potential pharmacodynamic marker. And really it's about neurodegeneration. As the cells die, we get this, this P75 extracellular domain released. Then we have this new predictive marker, biomarker of neuroinflammation called neopterin. And in the future, and it's one of the PhD students who's involved in actually refining the neopterin measurement, because as I said before, that this ELISA is not a sandwich ELISA, the commercial one, and because it's a very small molecule, it's actually extremely difficult to develop two monoclonals, and there's only really a polyclonal. So we've been developing this HPLC mass spectrometry method to measure neopterin. That's PhD student Vasilis Koronis, so yeah, that continues. So what about the future? What are we looking at now, and where we hope to be? So what about early biomarkers? If we just go back to this figure in Michael's recent publication, they've very nicely showed that you get a rise in NFL over this before the at the phenoconversion time, and then you get this steady state. Now, we know that progression markers don't behave like that, and other markers don't behave like that. And we also know that there are, we can't just rely on neurofilament light. We should be looking for a number of markers that can tell us about the progression of the disease over from diagnosis to death, about different pathological processes. So really, and so we need other biomarkers, and we also need to have. We think we need to have sort of a fingerprint, which means we need a lot of things that actually can give us an information about what's happening early and across the natural history. So that's what we'll be doing over the next few years, and identifying this early disease. We'll be so this is just a diagram just showing what we're planning to do. So what we have is we're looking firstly at preliminary data, which is early symptom, looking at some early symptoms by questionnaire, and looking at CSF and tears. This is a consortium of European and Australian and US neurologists and scientists, and so we also then be testing on pre-familial MND, things that we find, and then validating on a prospective cohort on a mixed population of pure motor symptoms and MND mimics to obtain this what we call a fingerprint. So in Australia, we will at Flinders and University of Tasmania, we'll be using high sensitivity single molecule array, or SMOA, and also at Flinders robotics assays, mass spectrometry, and ordinary ELISAs, just to validate our urinary biomarkers here and then here. Looking at their natural history, we'll also be looking at other biomarkers, that is CSF Chitin-1, which is actually a marker of microglial activation, and another wet biomarker, which is monocyte chemoattractin protein 1, which is associated with neuroinflammation. I think that we really need to decide whether this is a decent biomarker or not, because there's a number of studies which show that it actually may not change across progression and not be related to the ALF-SRS in blood, but maybe in CSF. So we really want to ask whether this is true. And basically, Paul Linger and other partners in our consortium will be measuring NFL, tau, phospho-tau, and also utilise proteomics to actually look for, especially in the CSF and in tears, to look for this early fingerprint. So what we're hoping to do is find an early fingerprint as a diagnostic predictor, and also a progression fingerprint as well, so we can have an early biomarker for pre-familial and early MND. So in summary, urinary P75 has been used in clinical trials. We've got this new predictive marker of pro-inflammation, and we plan to obtain a biomarker fingerprint of early MND and pre-familial MND across the natural history, and find additional prognostic markers, enable early treatment, and more information read pathological processes, and sort of make sure that we have this a good fingerprint and the natural history of the biomarkers that may hold promise. So I just want to acknowledge those people that were involved in all of the studies, especially the people with lived experience and the participants in our studies, and all the people in our lab, Stephanie and Vasilis and Tim and Megan and Danny, and especially from the M&D clinic, SA clinic, which is next door to our lab at the Flinders Medical Centre. David Schultz, Professor Dave Schultz, he's a great collaborator, and he's unfortunately couldn't come today because they have the clinic on Tuesdays. Our collaborators in US, Michael Benatar and Joan Wu, and Andrew Malaspina and Paul Lingor in Munich, and all the pre-Modi team, which is involved in this fingerprint, the fingerprint, M&D fingerprint analysis, which we're hoping to do over the next few years. Past members of the Neurotropic Lab, collaborators in Australia, especially Anna King and Jessica Collins, who put up with me last week, looking at their Samoa and funding. I'd like to acknowledge funding organisations, especially FightMND and MNDIA, NIH, and all these other funding bodies that were, and NHMRC, which are really have enabled this work. And happy to take any questions.
Great, thank you so much, Mary-Louise. Are there any questions from the floor? If not, I might just ask you one to get us started, Mary-Louise. Just wondering how you first went about identifying which biomarkers might be have potential as biomarkers for ALS.
Well, we first looked at we considered we were looking at the P75 in another respect, and we first of all looked in the SOD1 mouse model in the urine from SOD mice, and just to see whether we could identify the P75 as disease progressed in the SOD mouse, and then we moved on to humans. So we sort of just had this hypothesis because motor neurons are getting sick, that they are upregulating P75. The extracellular domain must be cleaved, and because it had been found in urine in rats that had an injury, we just hypothesised that it could be found in mice urine, SOD1 mice. Yeah. And yeah.
Okay, great. Just another question then, if there's no other from the floor. Given that there's been a lot of discussion in the field about how the challenges of using the ALS-FRS as a primary outcome measure for clinical trials, wondering whether you've looked at any other outcome measures for trials with regards to correlation with your neoptrin, for example, survival.
Yes, we have looked at survival, because that's really about prognosis, and I sort of we don't think neoptrin per se in our group. Don't forget, we only had 46 in our study. Yeah. It wasn't really. It was prognostic if we divided the patients up into high and low high and low neoptrin, but as a group, it wasn't prognostic in a Cox analysis. So whereas P75 was, and also the delta FRS, which is a change in the ALF-SRS per month, that was prognostic. But what it sort of tells us for the neoptrin is we need a lot larger study to see the prognostic value, because one other group had found that neoptrin was prognostic. So, but they had a larger cohort.
Okay, I might ask another question then. what do you see as the major barriers to biomarkers being integrated into clinical practice? For example, validation, accessibility, cost, a substantial enough panel. How do you see this going into practice?
I mean, at the moment, NFL it's expensive, but it has been I think more widely integrated than P75. But you have to remember that NFL's been around a lot longer than P75, 20, 30 years before they actually got a great assay. So we need to work on our assay, and not that our assay is poor, but for example, when NFL moved to the Samoa, they were suddenly being able to have more consistent measurements, and that's the sort of thing that we'd like to do, and like to make it more, and across the. I mean, I think what you're saying is it could be it hasn't really been integrated into all the lot of the trials. I mean, biomarkers are still sort of in the validation stage and stuff like that. Well, it does take a while for all of that to sort of come about. I think it's just the FDA process to go through the validation steps are a number of years.
We've just had a question in the chat that's come through. They just asked that, back to the previous question when we talked about markers correlating with neopterin. Did you notice any characteristics of the patients who had either high or low neopterin, for example, their age, sex, progression, anything like that?
The higher, looked like those that had the higher neopterin had a higher rate of progression. They were the faster progressors. But as I said, this is a small cohort. We need to extend that work. And what else was that, Sarah? I think it was Sarah.
Sarah, thank you for that question, Sarah.
So was that all she asked?
Just whether there were any characteristics that for example, age, sex, progression.
Yeah, look, it was faster rate, but there wasn't really any characteristics as in regards to age, because we actually took the effective age out, because we actually did not include those people that were over 72. Over 70 or 74 or something. So we basically precluded the older age group, because as you get into the 80s, people do have more and more neopterin. So, we had to preclude those people.
Thanks for that question, Sarah. I might just ask one more then, if there's nothing else from the floor. Can your biomarkers or any others that might be in development define heterogeneity? For example, disease onset, which types of cell death are involved, and the type of MND. And is this something that the biomarker field is directing towards in the future?
I think yes. To reduce heterogeneity, I think we can. You need to include a lot more than just NFL. So NFL and all of the biomarkers in more of a panel, and that will improve the ability to group people in regards to their disease, like those that have, for example, for the neopterin, those that are in more of a pro-inflammatory state. With the P75, those that are the higher progression rate would be more rapid disease for those, and so yeah, it should be able, they should, in theory, be able to use to group patients. So then you could use that group of patients to in specific trials, like for example the neopterin in anti-inflammatory, those that target an anti-inflammatory response.
There's no other questions from the floor. I think we might sort of wrap it up there. Right. Well, thank you so much, Mary-Louise, for such a fantastic talk and engaging us in a great discussion session at the end. So that'll do it for our November seminar and for the series for 2022. I'd just like to really thank everyone who tuned in during the launch of the FightMND Scientific Seminar Series this year. Our goal was really to provide researchers with the opportunity to hear from some of our national and international experts in the MND field and to hopefully identify potential areas for further collaborative research. So we hope you've really enjoyed this series for 2022, and we'll be returning with a fresh series of speakers in February 2023. So just in order to understand more about how the seminar series is supporting our scientific community, we will be sending out a short survey to attendees for feedback. This will only take a few minutes of your time and will allow you to help us shape the seminar series going forward. So please look out in your inbox in the coming days, and I really look forward to hearing some of your thoughts. So thank you again, Mary-Louise, and to everyone who tuned in today. We wish everyone a safe and happy holiday season, and we look forward to seeing you next year.
Thank you.
Thank you.